So thank you, Diana, and thanks to the organizers, especially Duncan, for the opportunity to present our recent work on TTX-sensitive sodium channels in menthol-sensitive neurons. And thanks to you who have stuck for the last session. So as you've heard already, and you know, DRG neurons initiate our senses of touch, thermoreception, pain, and itch. Now, one of the most remarkable features of these neurons is that they're both functionally and morphologically specialized to encode distinct features of complex sensory stimuli. And as you heard from Bruce yesterday, one of these functional distinctions is that different classes of DRG neurons have quite remarkably different intrinsic excitability properties, including their firing rates and their action potential shapes. So a fundamental question in somatosensory biology is how do these distinct classes of DRG neurons achieve this difference in intrinsic excitability? In my group, we approached this problem, and by we, I mean Theanne Griffith, who is an amazing postdoctoral fellow in the lab, by analyzing a transgenic mouse model made by Becky Seal's group. So this is a lineage marker for the VGLUT3 lineage, and as you can easily appreciate by looking at this immunostaining immunohistochemistry of a cryosection, some of these neurons have quite large cell bodies, whereas others have very tiny cell bodies. Now, we were interested in the VGLUT3 lineage because work from Becky's group and others had implicated these different lineage neurons in a variety of different sensory modalities. For example, VGLUT3 is expressed in CLTMRs, which are gentle touch receptors that have been implicated in affective components of touch. They're also, the lineage marker is expressed in these A-beta SA1 afferents that Bruce talked about yesterday and that my lab has studied for a long time that are involved in discriminative touch. A subclass of these is also has morphological endings of free nerve endings. Chu-Fu Ma's group has shown that. And so those are presumably involved in nociception. And finally, one paper suggested that TRPM8 positive neurons of this lineage are involved in oxaliplatin-induced cold allodynia. So as a starting point, Theanne used calcium imaging to ask whether some of this functional diversity is recapitulated in a dish. So she used calcium imaging, as you can see here, and then perfused on a panel of chemicals that activate different populations of sensory neurons. And what you can see is that some of these neurons, particularly these that have a little green circle, are activated by menthol. Very few are activated by chloroquine. Many neurons are activated by capsaicin, and then a whole bunch of neurons in this dish respond to high potassium-induced depolarization. So when Theanne quantified these responses across many experimental replicates, what she found, somewhat to our surprise, is that the entire population of menthol-sensitive neurons in the adult DRG fall within this VGLUT3 lineage. So here you can see in this pie chart that about 8% of the VGLUT3 lineage neurons, which are expressing TD tomato, respond to menthol. A little bit less than 10% respond to capsaicin, so those are putative nociceptors, and the vast majority, these respond only to high potassium. So we would think that these would probably include those mechanoreceptors that respond to touch stimuli. By contrast, when she looked at the non-VGLUT3 lineage, so those neurons that don't express TD tomato, she found that about half of them respond to capsaicin, so those we would think would be nociceptors, and then the other half responded only to high potassium. So Theanne set out to ask about differences in intrinsic excitability in these VGLUT3 lineage neurons, comparing those that respond to menthol to the other 90% of sensory neurons. So the first thing she asked is, does menthol evoke action potentials? And here you can see what the experiment looks like. So she's doing whole cell recordings from these very small diameter TD tomato positive neurons, and then perfusing on menthol. And what she sees is that indeed menthol evokes firing in these small diameter neurons, and it does so in a dose-dependent manner. So we get a modest amount of firing with 100 micromolar menthol and more firing with a millimolar. And that distinction is important because 1 millimolar menthol is reported to inhibit TRPA1 but activate TRPM8. So she proposed then that this evoked firing is through TRPM8, not TRPA1. Now, one thing that she noticed when she was recording from these neurons is that these menthol-sensitive, presumably TRPM8 expressing neurons, show a remarkable tonic excitability. So in the absence of stimuli at room temperature, she found that almost 90% of these menthol-sensitive neurons fired robust trains of action potentials. About half showed this sort of sustained firing pattern, whereas the other half showed about a phasic firing pattern. And because these neurons are grown at 37 degrees, but then recorded at 22, she hypothesized that TRPM8, which can be activated by room temperature, was providing the driving signal that gives rise to these action potentials. And she tested this by perfusing neurons with a TRPM8 inhibitor, PBMC, and found indeed that PBMC is sufficient to suppress this spontaneous or non-evoked firing. So she concluded then that these menthol-sensitive neurons fire TRPM8-dependent discharges at room temperature. And this sort of ongoing activity is quite unusual for DRG neurons that have not been subjected to an injury model. So she wanted to explore the basis of this excitability a bit more. As a first step, she did current clamp recordings and just analyzed some basic measures of excitability, including doing phase plot analysis, as you see here. And what she's comparing here, as I said, are the menthol-sensitive neurons to the menthol-insensitive neurons. And so what she found is that the menthol-sensitive neurons are sitting at a more depolarized membrane potential, and they have a lower threshold. So between the higher VM and the lower threshold, they're sitting about 10 millivolts closer to their action potential threshold than are the menthol-insensitive neurons. Moreover, she saw that they were firing a higher number of action potentials. This is in response to current injection, and I've already said they have higher firing rates. And then they showed a longer action potential duration. And the main reason they show this higher duration is that these neurons, as a population, are missing this shoulder that is quite characteristic of nociceptors, of small diameter nociceptors. And if you look at the menthol-insensitive neurons, you see that there is maybe a couple of populations, a population that also lacks the shoulder and a population that probably has the shoulder. And so I'm speculating here, but I would think that these potentially could be those CLTMRs, whereas these would be nociceptors. Now, yesterday, Bruce talked about some of the mechanisms that can give rise to different excitability. But one thing that we wanted to focus on, and Thean really chose to focus on first, is differences in voltage-gated sodium channels in these populations. So here I'm summarizing data from a number of groups and from a recent review out of Steve Waxman's lab that summarizes the expression of different voltage-activated sodium channels in A fibers versus C fibers. And what you can see is that C fibers, these small diameter neurons, express a number of NAV subunits. So Thean took a pharmacological approach to ask whether any of these NAV subunits or currents through these channels are required for action potential firing in these neurons that I've described. So the first thing she used is TTX. So what she found, quite to our surprise, is that TTX almost completely abolishes action potential firing in these small diameter menthol-sensitive neurons. By contrast, TTX had little effect on the menthol-insensitive neuron population, although again, you can see some spread to the data here, indicating that there's definitely some diversity in these menthol-insensitive VGLUT3 lineage neurons. So These data indicate that the TTX-resistant sodium channels, NAV 1.8 and 1.9, are not essential for driving action potential firing in the menthol-sensitive neurons under these experimental conditions. So we next turn to NAV 1.7, and we used a couple of different inhibitors, the 771, the Pfizer compound, as well as a spider toxin, PN3A, that was generously provided by Irina Vetner. And so what we found is that these NAV 1.7 inhibitors had essentially no effect on menthol-sensitive action potential or on evoked firing in these menthol-sensitive neurons, and to our surprise, had really no significant effect on the menthol-insensitive neurons as well. But again, there is some diversity there. There's some variability in that population. So that indicates that NAV 1.7 is not driving spike firing under these conditions. So we next turn to NAV 1.1. So we used ICA, which is a selective inhibitor of NAV 1.1 and 1.3. And we found that ICA at 500 nanomolar was quite effective at suppressing action potential firing. So we see that firing rates were reduced by about 80% by this compound. By contrast, ICA had little effect on the menthol-insensitive population. So that suggests that NAV 1.1 is important for spike firing in these neurons in vitro. Now, one of the reasons that Thean wanted to analyze sodium channels was inspired really by some work done near the turn of the century by Bruce and his colleagues, showing that slow inactivation of TTX-resistant sodium channels contributes to action potential adaptation in nociceptors. So Thean got very excited about these data and proposed the converse hypothesis. She thought that perhaps NAV TTX-sensitive sodium channels may be resistant to slow inactivation, thus making them available for driving spike firing in the menthol-sensitive neurons. So she tested this hypothesis using voltage clamp recordings, and what you can see here is a comparison of entry into slow, the kinetics of entry into slow inactivation. Sorry, I'm having trouble with this pointer. In the menthol-sensitive neurons versus menthol-insensitive neurons. And what you can immediately appreciate is that these neurons are quite resistant to entry into slow inactivation. So they have a dramatically prolonged time constant compared to menthol-insensitive neurons. Similarly, they recover faster from slow inactivation compared to menthol-insensitive neurons. Now, what you're probably asking is whether NAV 1.1 is responsible for these differences that she observed. So to test this, Thean repeated the experiment in the presence of ICA, and then by doing current subtraction, she first estimated the fraction of total sodium current carried by NAV 1.1, and what she found is that NAV 1.1, or rather the ICA-sensitive component of the sodium current, represents only about 38% of the total current. But remember, I told you that this is responsible for driving about 80% of the action potential firing. So when she looked at the kinetics of inactivation after application of ICA, she saw that in the menthol-sensitive neurons, the entry into slow inactivation is now comparable to what she saw in the menthol-insensitive neuron population. And similarly, the recovery from inactivation is prolonged after ICA. So together, these data bring us to the working model that I'm showing you here. We propose that in adult DRG neurons, menthol activates TRPM8 to depolarize the sensory neuron. That then activates voltage-gated sodium channels, including NAV 1.1, which then cycles through a fast activated state to support high-frequency firing in these neurons. Of course, this leads to more questions than answers, and some of the questions that Thean is interested in pursuing include the ones I've summarized here. So she's shown that menthol-sensitive neurons are a highly excitable DRG neuron population that mediates persistent action potential firing at room temperature, at least in vitro. And so yesterday, Bruce told us that context matters. And so one thing we're really interested in finding out is what is the context for these NAV 1.1 channels. So Felix Viana and others have shown that KV1s, HCNs, and 2-pore potassium channels are important for setting cold thresholds in nociceptors. And so we're interested to know what are the contributions. What are the potassium channels in these menthol-sensitive neurons, and what is their contributions to this intrinsic excitability? We've shown that NAV 1.1 and perhaps other TTX-sensitive sodium channels dominate action potential firing in menthol-sensitive neurons. And so a key question is whether animals harboring loss of function mutations in NAV 1.1 have deficits in cold or menthol-sensitive sensing. And we've now made a peripheral neuron-specific knockout of SCN1A to test that hypothesis. And of course, we were unable to use pharmacology to assess the contribution of 1.6, but we're very interested in understanding whether NAV 1.6 is contributing to excitability. We found that the NAV slow inactivation kinetics differ between menthol-sensitive neurons and other small diameter neurons, and we're really interested to know whether beta subunits are playing a role in this difference in kinetics. And finally, we're excited about the therapeutic implications of these results, and we're quite interested to know whether NAV 1.1 directed therapeutics might have efficacy in cold allodynia, particularly in CIPN models. So I'd like to acknowledge those who contributed to the work. All the data I showed you today is the work of one very tenacious and talented postdoc, Theanne Griffith, who's pictured here. Theanne is on the job market. She's looking for tenure-track positions, and I encourage you to recruit her if your department has any openings. All of this work was started as coming out of kind of an interesting observation during the electrophysiology section of the MBL neurobiology course, where Theanne was my TA a few years ago. So I want to encourage you to think about sending your students and postdocs to the MBL neurobiology course. Next year is our 50th anniversary. We're going to have a party, also called a symposium, on July 18th. So I encourage you to mark the date on your calendar. Think about, if you've been involved in the course, think about coming to the anniversary celebration, and encourage your graduate students and postdoctoral fellows to apply. I will thank you for your attention and take any questions.
Thank you very much. So, questions? Bruce.
Very cool. Is there any information from previous studies on axon conduction or with skin nerve preparations, whether the menthol-sensitive neurons will fire at a higher frequency generally than other C-type fibers?
Thank you for that question. Felix's group has published that with skin nerve preparation, and in fact, they do sit and show these phasic and tonic bursts, high-frequency bursts in skin nerve preparations, very similar to what we see in vitro. And I would say that initially, when we were doing these recordings at Woods Hole, we were throwing away all those neurons because we thought they were all unhealthy. We finally figured out that actually they weren't unhealthy. They would just sit and putter along for, tens of minutes.
Yes.
There's been many publications on RNA sequencing trying to bucket different populations of DRG neurons. Are those consistent with what you've shown here in terms of, is trip and mate co-expressed with NAV 1.1 from RNA sequencing experiments?
Yeah, NAV 1.1 is broadly expressed in the adult DRG, and I didn't show it here, but we published this in the paper. We did RNA scope with trip and mate in the TD tomato mice and looked at all of those sodium channel subunits that I showed you, and they're all expressed in the trip and mate population. So there's this real disconnect between message and function in DRG neurons, with the caveat, of course, that these are DRG neurons in a dish, not in vivo.
Do Dravet patients with loss of functions in SCN1A, do they show changes in cold sensitivity?
It's a great question. I haven't been able to find any information on that in the literature. But it's something that I think would be really important to look at. SCN1A is also, there's a familial form of migraine in an SCN1A patients. But again, I found nothing specific about cold sensitivity.
What do you suppose the 60% of the sodium channel current that doesn't contribute to action potential firing is what do you suppose all of the other sodium current is there for? It's not 1.1 and not
I think that's a great question. I mean, I think what we really need to do here is action potential clamp, because what I showed you with the 70% of current is in response to a step depolarization, right? And so there is this can be a real disconnect between the currents flowing during an action potential and those in response to a step.
Ellen, I have a question. In one of your last points was like, what's the function of this set of neurons that appear to have relatively high level of excitability? And if I'm remembering correctly, 1.1 is downregulated during development. And so I'm wondering whether you have looked to see whether there's higher levels of expression early on and maybe downregulation later on.
We have not done any kind of expression analysis through the course of development. It's a great question. We've only looked in adults. Okay.
Great. Well, thank you so much. Steve.